Welcome everybody to today's SPHN webinar on the medical imaging concepts, the latest extension of the SPHN interoperability framework. We're happy to see that this relevant topic has sparked so much interest. We're presenting Imaging Data Fair, insights on the new medical imaging extension of the SPHN semantic interoperability framework, presented by Dr. Edwin Voert. One of the key goals of SPHN is advancing the interoperability of health-relevant data for research and reuse. Such data is highly diverse, be it clinical routine data, but also data from clinical trials, registries, patient-reported outcome measures, or sequencing facilities, just to name a few. Data interoperability means that each data point has a clearly defined meaning that can be interpreted unambiguously and consistently. Interoperable data is understood alike across institutions and projects. It's ideally aligned nationally or even internationally. It should also be meaningful over time, and it's understood by both humans and machines. And this allows data-driven insights, for example, by machine learning algorithms or artificial intelligence. Such data interoperability is achieved by the use of controlled vocabulary, international standards, like, for example, SNOMED-CT for clinical terms, LOINC for laboratory results, or, as we will see, DICOM for imaging data. Interoperability is also promoted by the use of semantically well-defined building blocks representing specific aspects of the data. These building blocks or concepts are aligned in a data schema like the SPHN schema across user groups. The SPHN schema has evolved step by step since 2019 and has already covered a broad range of domains, including administrative items, demographics, medications and diagnosis, procedures and measurements, special concepts for oncology, microbiology, and many more. Medical imaging was already present, but with a single concept only, and thus largely underrepresented. Given the growing importance of medical imaging and the wealth of data it constitutes, the task was to structure the different aspects of imaging data, including procedures, formats, reagents, devices, and incorporate it into the existing SPHN schema. It was very encouraging to see that all the different stakeholders, including the hospitals, have committed to tackle this task together. With this, I would like to warmly welcome today's speaker, Dr. Edwin Voert from the University of Zurich. Edwin has a background in software engineering, biomedical engineering, and also holds a PhD in medical sciences. Already as a doctoral researcher in cancer research, he used advanced imaging techniques and data like PET or CT scans. But Edwin has also gained experience as a postdoctoral researcher at the Department of Nuclear Medicine at the University Hospital Zurich and has worked at the Biomedical Image Computing Group at the ETH Zurich. He's currently working at the Department of Quantitative Biomedicine at the University of Zurich on the Biomedical Informatics Imaging Platform Project, BMI2, from the Loop Zurich. I list all these to emphasize how fortunate SPHN was to have such a qualified imaging expert as Edwin on board for this project. Over the past 2 years, we have worked very closely with Edwin to align the medical imaging extension with all its concepts seamlessly with the existing SPHN schema, and this has always been very constructive. It's a pleasure to see the results of this productive collaboration presented as a whole today. Very much looking forward to your talk, Edwin.
Okay, thank you very much for the introduction. Yes, so my name is Edwin Voert, and I'm going to present this now. So we start with the introduction, then we have something about the DICOM standard. We talk a little bit about the SPHN medical imaging concepts as an overview. We talk a little bit about the documentation, and then we focus on some more details. So as Harold already kind of mentioned, that expert analysis and interpretation of clinical image data is critical in the diagnostic and precision of treatment. During the past 25 years or so, hospitals and research institutions have stored and archived huge amounts of medical imaging data. So the medical imaging research data provides an invaluable data source that can potentially be used to improve patient diagnosis, treatment, procedures, but also better algorithms for medical scanning, automated diagnosis, outcome predictions, etc. Developing state-of-art methods and algorithms in Switzerland requires the use of large data sets obtained in diverse populations and in multiple hospitals and research institutions across the country. Many challenges around the fair use, reuse, and integration of these medical imaging research data sets remained unresolved. Yeah. The medical imaging concepts in this new release of SPHN marks a major milestone in addressing these issues. So yes, as Harold already mentioned, there's a kind of there was a previous SPHN there was a previous imaging procedure in the SPHN semantic framework. It had, for example, it could indicate the body side, start and a stop date. There was an intent, and of course the kind of procedure it was performed, but many details were kind of missing. So we sent out a questionnaire to several researchers in the field. So how do you, or would you like to search for interesting medical imaging data sets? So the clinical researchers responded mainly with they want to search for patient demographics, diagnosis, disease, anatomical location, imaging procedures, and then, of course, the technical researchers want to have a little bit more technical details, so the imaging protocols, scan parameters, maybe traces and contrast agents. And we made a selection of the most important and commonly requested data items. And we looked also at the metadata described for imaging in the DICOM standard. So what data was already available. So then a little bit about the DICOM standards. So most medical imaging data, like for example, X-rays and PET and CT, in hospitals is stored and transferred in accordance with the DICOM standards. So the DICOM is the international standard for medical images and related information. NEMA holds the copyright to the published standards. And DICOM is already a little bit older, so it was first published in 1993. And since then, there were, of course, many additions, deletions, and changes. So you should always look for the current version in the URL if you are looking for something in the DICOM standard. The DICOM standard is sometimes a little bit difficult to browse, but Analytics has a nice browser for the DICOM modalities. It's shown here, which you can basically find kind of the modalities. You can click on it and you can find more information. You can also click on the right side. There's a view in the standard, which then links to the real DICOM standard. So DICOM standard is huge. It describes all kinds of things from storage to network communications in a total of 22 parts. You see them also on the right side. So DICOM part 1 until 22. Part 3 is basically the information object definitions where we are interested in. So these new SPHN imaging concepts are built upon these image information object definitions in the DICOM standard. Data providers can also use the DICOM metadata as a data source for instances of the SPHN concepts. And many, but not all DICOM terms, which is called the DCM terminology, are provided in RDF and are also available in the SPHN terminology service. I don't know, maybe you may know already the terminology service from SPHN. Otherwise, you can find the links down here, and you can see here that it's already available. And then a little bit about the DICOM standard imaging entity relationship model. So that's shown on the left side. And on the right side, you see a typical workstation or console where images are stored or on a scanner, for example. Usually there's a list of exams like here. And the exam is performed on a patient. That's usually listed here. So you can select an exam for a specific patient on a specific date. And here you see the modalities. Then it pops up a couple of series here. So during this exam, we could, for example, have performed a localizer on MR. It could be a calibration scan on MR. There could be a PET stress scan on a PET scanner in this series. And if I would click on one of these series, it would list all the images that are obtained in the series. So it could be, let's say, 400 or so, or whatever you wanted. So you can see this corresponds a little bit. So the exam corresponds to the entity relationship model from the DICOM. So this is called study. So the name here corresponds to a patient. So a patient is a subject of a study, and a study contains series, and the series corresponds to this one here. And there's also series can contain many different things, can plants, dose, waveforms, and all kinds of things in the standard, but we are interested in the images that you find down here. And the series was created by some equipment, for example, a PET-CT scanner or a MR scanner. So DICOM images are usually stored on a picture archiving and communication system, also called PACS. DICOM images can also be stored in files, and whatever you choose, there are basically two kinds of formats to store these images. So there's the legacy single frame format. Here, every 2D image slice or frame is stored in a single file containing all the metadata in a header and the image pixel data in the remaining part of the file. So you have file 1, this is the header data. Here you have the pixel data. You have file 2, almost the same header, other pixel data. File 3, almost the same header with the next slides, basically pixel data here. So the disadvantage of the single format is that many data items in the header are optional, so that's not so good for us. It means that many can be empty. It is also the most common way DICOM images are stored because of, it's used already for a very long time. And the other disadvantage, of course, that each file has almost an exact copy of the same data, so it's not very efficient. So the newer version is an enhanced multi-frame format, where you have one shared header, which contains all the standard header information. Then you have the pixel data stored in the same file for different slices. So here you have for frame 1, here you have the pixel data for frame 2, 3, and 4. And the specific details for pixel frame 1 are in a per frame header data that can be listed here as part of the header. So the good thing for us is that many data items are mandatory. So you can have you can extract a lot of information from this DICOM files. The problem is it's only getting slowly accepted and applied. And the other advantage, of course, that all data is in one file, which makes reading of these files way faster. So if I go back to the standard browser, to the DICOM browser, you see that we have a list of CIODs. So these are composite information object definitions. That is something like the modality in kind of other words. It describes in modules all the metadata for a specific modality. So there can be, as I said, two different versions. There's the normal MR image, for example, here, and there's the enhanced MR type here on the other side. So the CIODs consists of several modules. And here you can also see that this one has an enhanced type of module in here, and it has multi-frame functional groups where specific metadata for a specific frame to the slice is stored. If I look in the DICOM header, on the DICOM definition, the official NEMA definitions, I see basically something like this. So you have the real-world objects like patient, study, series, which then contain modules, patients, information, and general study information, the patient study module. You can also look at this with the standard DICOM browser again. So here you see the CIODs. In there, if you open them, you see the patient modules, the clinical trial subject modules, and you also have the patient study module. And in these modules are usually a list of DICOM data elements, or sometimes attributes, and sometimes they call them also tags. And these basically describe the data, the metadata. So an attribute, a data element, is composed of the following parts. There's a tag, so this is the actual real tag, so it has this format, which is shown here as well. It has a type, so if it's optional or required or whatever. It has a keyword, so this human readable version of the tag, basically. It has value multiplicity, which indicates how many how many values that can be assigned to this tag or attribute. It has a value representation like H string. This indicates basically the data format. And a special type of attribute is the code string. Here you can have DICOM defines all kinds of enumerated values. For example, if you look here, this is then the smoking status. The smoking status can have enumerated values, yes, no, or unknown. And these are all defined in the DICOM documents itself and not in the terminology. So 001 means not pregnant, for example. F means female, L means left, yes means yes, but only for this specific tag in this modality. Another special one is the sequence. So this kind of a nested attribute. Here you can, for example, store, or here are the terminology terms stored, for example, SNOMED terminology terms, or the DICOM terminology terms, or the UCOM. And the values that are allowed in these terms are usually listed in a context ID, in context ID tables. So if we again look, for example, at a general image module, in there we see the atomic region sequence. So this one is nested attributes. And one of these nested attributes is again a sequence, and in there can be another sequence, for example, here. You also see that it has a code value, code scheme designator, a code scheme version, and a code meaning, which refer basically to the table, anatomical modifier, for example, where you can have your SNOMED code scheme with the code values and the code meaning. And these correspond then to these tags as well. There's also a DCM option with the code and the code meaning. Note that the code meaning of SNOMED, so that's what's written here, can be slightly different than what's written in the SNOMED standard. And this is how it looks in the DICOM standard, so the official NEMA version. So you have here the attribute names, the tag, the types, and a description. An important thing is here is the larger than symbol, which means that everything that's listed in this table here, entry, is nested inside this attribute. You can also have the double or the triple nested attributes listed here. So then the SPHN medical concepts and an overview. So on the left side, again, you see the DICOM entity relationship diagram. So the patient has maybe a visit, the patient is part of a study, so the exam. And the study has series, and the series is images, and the images are stored in some kind of DICOM files. Now for SPHN, we created something similar. The patient is then called, of course, the subject pseudo-identifier. We cannot have patient names in there. The visit is something like an administrative case. The study is then called imaging procedure in our case, which inherits from medical procedure. The series is in our case called imaging series, and we can have different subclasses of imaging series. For example, MR imaging series, CT imaging series, PET imaging series, and X-ray imaging series. And we can add multiple other ones in the future if there is need for it. And of course, imaging series has a group of imaging frames, which correspond to the images in the DICOM standard. And they, of course, have the same subclasses. So the MR imaging series has the MR imaging frame. So these ones already existed in SPHN. Imaging procedure also existed, but it's now extended. The SPHN data file, for example, is on the imaging series level. So you could link to a folder or a DICOM there, or whatever you want to, where that has the real pixel data and the real DICOM files. As said, more imaging modalities could be added. And the imaging frame is a little bit special because if you have, for example, 200 image slices in a series, you don't have to specify all 400 imaging frames because they all contain almost the same header data. And that's why you only need one imaging frame for a series, unless some properties change. For example, if you scan the head with part of the lung, for example, then you have two different locations. Then you can add, for example, two imaging frames indicating one for the head and one for the lung. So another important part is the documentation. So there's, of course, the SPHN Schema scope, which shows here, for example, the imaging procedure and all connected concepts. You can see that there are many new ones, but there are also many existing ones. And there's, of course, the SPHN GitLab. So you can find it here. It lists these folders here. So there's a data set which contains the SPHN release in Excel format and some documentation files as well. There's a documentation folder that has the pilot HTML documentation. So that's the part where you can nicely click through the documentation and get some information. It also has the quality insurance, so the SHACL and the Sparkle files. It has the RDF schema in there. So that's the turtle and the OAL format. It has the templates if you want to add new concepts to it, for example, and it has a license and a README. So if we look at one document, for example, then we find in there's a chapter called Rationale, which globally describes why we would need this and what's the purpose of this new concepts. There's a chapter called Comparison to Other Standards, where we compare the design of what we developed to, for example, other standards, like, for example, the DICOM standard. There's a chapter called New Concept Information, where you find all the information about the concept. It's a little bit the same as the Excel file. So here's the concept name. There's the concept type is this. The concept name is here. The general description is here. The contextualized conception name is here. We have, for example, the type, the standard, value sets, meaning binding, and everything is in there. So these are all the inherited composed of. Here is a new composed of listed. And here you can find basically all the metadata descriptions. Then there's a chapter called Impact on the SPHN dataset. So in this case, it has there's nothing special, but sometimes there can be an impact on the existing dataset. A very important chapter is also the discussion. So if we encounter problems or difficulties, then we discuss it here. We provide some solutions. We also discuss design, decisions we made, etc. Then there's also an example chapter where we list the related DICOM tags. So for example, if this code is your composed of in the procedure, I think, then we say you can find the code, for example, in this DICOM attribute or tag, so the procedure code sequence. This probably has the value you're looking for your composed of code. It is available in both the legacy and enhanced DICOM. It is available for these modalities. We provide a link to the DICOM standard, which contains a description, and we also provide a link to the CID table, which contains all the terms that are allowed for this DICOM code sequence. We do this for, of course, many composed offs. We also sometimes provide conversions to, for example, SNOMED CT. So if you have, for example, DICOM pregnancy status, the DICOM terms would be 0001, 2, 3, and 4. So which means then not pregnant, and then we propose SNOMED CT code for your subject pregnancy status code. That would then be this code, for example, not pregnant. Yes, of course, a better future solution would be that DICOM just replaces these kind of old codes with officially DICOM or SNOMED CT terminology codes. But so far, it's not done yet. Another example is, for example, the imaging procedure code. So the imaging procedure code is often not provided in DICOM directly. So as an alternative, we, for example, propose to use the modality attribute tag or the SOP class UID attribute or tag. So it's, for example, listed here. Then you can, if the modality, for example, says it is CT, computed tomography, then you could add the proposed code to imaging procedure. For the imaging procedure code, you could use this SNOMED CT code in there. And you could do the same, for example, for the SOP class UID, this which also describes what type of scan it is as a CT image. And you can use this table again to find the SNOMED CT code. So then a little bit more to the details of the design. So we have this imaging procedure here. It was already there, but we extended it. It links to, for example, administrative case, which is not always necessary to have. For example, in ReachS scans, this is probably not available there. Of course, the imaging procedure also links to the imaging series. Here you can add all your imaging concepts. The imaging procedure also has a code. And as said before, the proposed DICOM tag procedure code sequence is not very often provided, but you can then use, for example, the modality as an alternative. If the DCN code of modality says computed tomography, then you can use the SNOMED CT computed tomography procedure as the code here. The body site is specified on 2 levels. So it's on the imaging procedure level and also on the lower imaging frame level. So here you could do 2 things. You can make a list of all body sites you have at a lower level. And you can add all those body sites here and make a unique list of, so then you know this scan has all these body sites included. Or you could, for example, specify a higher level body site. So for example, if the body site at the lower level is head, neck, chest, abdomen, pelvis, and limbs, then you could add here, this is a whole body scan. For the text descriptions, it is preferable to have structured text and in English. And it's also not always the case, but yeah, it's the preferred versions. So then we have a lot of parameters like subject age, sex, weight, height, BMI, and pregnancy status, which are extracted from can be extracted from the DICOM. These are not only the properties of a subject, but they are also scanning parameters. So the scanner, for example, knows by using these parameters how much energy the scanner can put into a body. And there's another catch. So body weight and body height are usually entered at the scanner console, but they don't have to be very precise. It can be a fair estimate by a clinician. So when you enter the scanner room, the clinician looks at you and says, oh, you're 1 meter 80, your weight 80 kilos, and he or she enters these numbers on the console, and they end up like that in the DICOM header. So be careful with those numbers. It can be a good estimate, but they don't have to be very accurate. Then we have, of course, the imaging series concept, which again has the administrative case, which is not always defined for research scans. There's again free text fields like a protocol name and descriptions in there. They should preferably be in English and structured. We can attach, of course, all the imaging frames, so all the 2D images that are scanned in an imaging series. And as said before, we don't want an imaging frame for every single 2D slice, but only the unique ones. So with different parameters in there. So if you have 400 scans, you have 400 image slices, you end up maybe with 1 or 2 imaging frame concepts attached to the imaging series. Unless you have a very specific and you want to really have every image frame specified. We can also specify the imaging device. The imaging device is different than a single modality. An imaging device can, for example, be a PET-CT scanner, a PET-MR scanner, a SPECT-CT, and we will explain it a little bit more in the next slides. We can have the data file. It's specified in the image series, as also said before. You can specify a folder or a DICOM there, so the index file or a folder tree. Imaging series has a start date time and an end date time. The end date time is usually not given in DICOM, but the start date time is. But it's usually distributed in 2 tags. So there's a start date and a start time in 2 different attributes. So you have to combine these into a new date time. And here you can also add the modality. So every imaging series is about 1 modality. So you can either have a CT modality or the PET modality or the MR modality or anything else specified under the SNOMED. Then we can also have the imaging series as a quantity, so the number of frames, so the number of 2D slices in this imaging series. For example, it can be 400 imaging slices, while you have only 1 or 2 imaging frames specified. So the imaging series also have descendants for MRI, CT, PET, and X-ray. They are listed for here. So one additional thing these descendants have is body position. So the SPHN body position is maybe a little bit different than from what you're used to from DICOM. In DICOM, we, for example, specify that the patient lies head first supine in the scanner. It can be, for example, look like this. So if the scanner is here, the patient lies on his back with his head first in the scanner. So this results then in 2 terms of body position, one with SNOMED CT code head first, and one supine body position. So this one DICOM term is basically split into 2 SNOMED CT codes. You can also read the documentation for it. It explains it a little bit more. Then, of course, if you have the image frame for computed tomography, then the image frame should also be of type computed tomography. And if you have the MR, then you should also use the MR imaging frame here, of course, etc. Then a little bit more about the imaging device. So the imaging device has a type code, so that indicates the type of scanner, so PET-CT, for example. But DICOM does not have this, does not have a metadata for this. So, but it has a manufacturer, it has a manufacturer model name, and it has a device serial number in the DICOM header. And these 3 together should uniquely identify a scanner in a hospital. So a hospital could, for example, make a lookup table where you have these 3 values, and which then gives you a type of scanner. For example, if this is a Siemens, some kind of modeling device, then you know it is a PET-CT scanner or a PET-MR scanner. So you can add then this code to type code here. Another problem is that in SNOMED-CT, not every scanner is a descendant of the clinical equipment or device. There is also a possibility that your scanner is on the device systems. So we have changed the parent medical device to include both options. So your code has to be descendant of this one or this one. But a future solution would, of course, be that we ask SNOMED to update the terminology to have all these scanners under a close common root. But for now, it's not like that, so we have these two. We can also attach software, the SPHN software, and it has cardinality 0 to n, so you can add multiple software concepts. This is usually the case for complex scanners. When you have a PET-CT, then you have usually two types of software on it. There's also, of course, the manufacturer name and the module name if you want to search for only Siemens scanners or only Philips scanners or whatever scanners, or a specific model. Then we come to imaging frame, which is basically your 2D description of your 2D image. It has an defined anatomical projection, so basically which plane is the image. It can be, for example, sagittal plane, a coronal plane, axial plane, or any other plane. The imaging frame also specifies the imaging metric. So the imaging metric is basically the physical quantity that was measured or calculated. So if you could, for example, be T1 or T2 relaxation time. So it's basically the gray value. What is the meaning of the gray values in your image? So for a T1 scan, you have T1 relaxation time. So then this would be T1. You could also have a diffusion scan, then it would be diffusion coefficient, or it could be linear attenuation coefficients, or radio density enhanced fields, or standard uptake values, or whatever you have. And you can specify it in the imaging metric. Then, of course, we have the image dimensions there that will be explained next. There's also an imaging frame. And the imaging frame is a little bit a complicated text. It was also explained in the next part, but it specifies the important characteristics of the image. Here again, we have the body side now at the lower level, so you can specify that it's a head or neck or chest scan. It would, of course, be better to add more details like the heart, lung, kidney, or liver, or prostate, or whatever you scanned. And you can also specify the content qualification. So if the scan was a product, research, or service, so product means it's approved for clinical use. If it was a research scan, then it's only for research purposes, or service, then it's only for calibration purposes or whatever service was done. Then you can provide a data compression algorithm details. So if the data was, for example, compressed using a lossy data compression algorithm, then you can specify the algorithm and also the compression factor. So the DICOM text, image type, and image frame are more or less the same, but this one is on the image level, and this is a little bit more on the frame level. But the content is the same. It's a kind of ancient tag where all kinds of information, vendor private terms, for example, could and is often ended by vendors. So the first, for example, look here, you see that it's the multi-value tag. It has 2 or more values. Value 1 and 2 are very well defined. It can only be these 3 values here, and value 2 can only be this ones too. Value 3 and more and others can be specified, don't have to be specified. Vendors can add their own private terms, and the meaning of those terms usually remains unclear and could change for each modality scanner and software. So that's a little bit difficult. So we created a value set containing only the DICOM defined terms. You find them on the right side. And in the future, we could add more when needed. The DICOM terms have here a specific order, so you have value 1, value 2, value 3, etc. But we add them each term separately to the imaging frame concept. These terms can also be used in other as information source for other concepts. For example, the T1 could be used in the imaging metric concept. So now quickly to image dimensions. So if you have a 2D image, like this one, where you define the pixels, and this is the center of the pixel, then you can have a number of columns defined, so the number of pixels in this direction, the number of rows in this direction. You can have the distance between these pixels from the center to center. You can have the distance in the column space. You can have slice thickness in a 2D stack, for example. You can have the slice spacing, so from the center from the slice to the next center of slice, and you can specify all these in the image dimensions and in the pixel dimensions concepts. And the sizes are in the patients, so distances in the patients and not detector sizes. Then we have contrast agents, phase and contrast agent administration events that can be important as well. You can attach them here. So the contrast agent administration event inherits from the existing SPHN drug administration event. Contrast agents inherit from the existing SPHN drug. The contrast agent activity ingredient inherits from the existing SPHN substance. And we have a new one, flow profile interval. So the contrast agent is listed here. You can specify a contrast agent, which is then described here, and which has an active ingredient that's specified here. And you can add, for example, a profile. And there's the profile from an automated or a programmable infusion pump. So the route is usually intravenous, which you can specify here. The drug article concept, the data for data is not always available. Usually there's a name in the DICOM. The GTIN code is usually not provided, but can maybe be found in other hospital records or patient records, or you can maybe found using a lookup table. The quantity here specified is the total administered volume, so the diluted contrast agents. Then we have in the contrast agent, we have the real active ingredient. You can specify it as a SNOMED, as a descendant of the SNOMED contrast media, for example, this one. Then you can specify the concentration, so 469, for example, here, milligrams per milliliter. You can specify the total administered amount of the undiluted contrast agents, and sometimes you can specify the volume percentage. As said, the contrast agent is usually provided with an infusion pump. And you can specify the flow profile of this infusion pump. For example, you have a start time and a stop time. And during this interval, the flow rate was, for example, 0.5 milliliter per second for a duration of 5 seconds. And then the total volume can be 2.5 milliliters. This can all be specified here. There can be a second interval connected as well with slightly different properties, for example. Then we have something like contrast agent phase, and this basically specifies where the contrast is at the time the image was recorded by the scanner. So you can have a pre-contrast image, then there's no contrast in this image slice there. You can wait a few seconds, and then the contrast agent distributes in the arteries of your body. And then you have, for example, a late arterial phase where arteries are kind of highlighted. You can have a portal venous phase or a delayed phase, which is usually after several minutes of uptake. And all these different phases you can specify here. Then we have the positron emission tomography imaging frame, which has the radiopharmaceutical administration event. So it's a little bit the same as for contrast agents, but now about radiopharmaceutical administrations. So again, the radiopharmaceutical administration event inherits from the SPHN drug administration event. The radiopharmaceutical inherits from the SPHN drug. The radionuclide inherits from the SPHN substance. And we have a new one that's concept is called radioactivity. Let's go a little bit more quickly. So the radiopharmaceutical administration event has a radiopharmaceutical, and the radiopharmaceutical has a specific quantity. That's the amount of radiopharmaceutical in cubic centimeters. It can have, for example, active ingredients, for example, 18F FDG. And the radionuclide can be specified separately. And it's also called radioactive nuclei, radioisotope, or radioactive isotope. It can, for example, be the 18F. You can specify a quantity, but it's usually not provided in DICOM. DICOM provides a half-life, so it's the time, characteristic time for the decay curve, like this. And DICOM also specifies a positron fraction, which is important for the PET scanners. And there's a radioactivity concept which specifies the activity at a specific time point. So as activity decays, it's important to also record at which time the activity was measured. In DICOM, it's usually the start of the contrast, the start of the radiopharmaceutical injection. Then the physiological state says something about the patient state. So is it patient in resting state, or did he get medication to induce a cardiac stress state? And that's also, again, the re-injection state, the reducible state, which kind of says where the radiopharmaceutical is located. Then we have also a synchronization concepts. Synchronizations have two composed of a method and signal source. And synchronization can, for example, be a cardiac synchronization or respiratory synchronization. So it can indicate the triggering during breathing, or an ECG trigger. So you can specify which signal was used and which method was used to register your image. But then we have several smaller ones. So there's, for example, the clinical trial study concept, which an image procedure can be part of. So of a clinical trial, so you can specify the clinical trial there. There's an imaging metric concept, which we kind of explained before. It's the T1 and the T2 values, the gray values. There's an anatomical projection concept. Here, it has a code and has a modifier, so you can see which slice or how the slice is oriented. Then there's an algorithm concept. There's a general one. And a child concept is a data compression algorithm, where you can specify which data compression type is used, the method and the ratio, and some other text descriptions like name and a resource locator. This is very much like the software concept that's already in SPHN. So as a summary, so we extended the SPHN concepts with the medical imaging concepts. So that means that we added or changed approximately 32 new concepts. And this allows for, hopefully, better description of the medical images, and easier search for interesting imaging data sets, and hopefully, in the end, more advanced research. I would recommend to carefully read documentation if you're going to use it. It contains a lot of useful information, conversions, and all kinds of difficulties and solutions we encountered to help you with this. For the future, there's also still some work to be done. We have to add more terms to the terminology, or we have to make We have to add more terminologies to the DICOM standard or the SNOMED CT standard, or at least update them so it fits more to the general needs. The DICOM standard defines many attributes that can be used as a metadata source, but in the legacy DICOM, only a few are mandatory, so there can be many empty or not provided at all. And several attributes are free text fields that can contain any text in any language, which has no structured information, so we should find a solution for this as well. And for this, basically, we started now a new project that tries to enhance the DICOM metadata. And we use the pixel data and other hospital sources, and use large language models to try to improve this kind of information. So that was it. Thank you very much for your attention. I would like to thank the university hospitals, universities, of Zurich, ETH Zurich, PHRT, the Loop Zurich, of course the SPHN teams, Harald Witte and Vasundra Tour and Sabine Westerday, and of course all contributors